Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The intricate architecture of the nervous system, a realm once shrouded in mystery, found a masterful cartographer in Santiago, Ramón y Cajal. His groundbreaking investigations, culminating in the publication of Texture of the Nervous System of Man and Vertebrates in 1889, illuminated the fundamental organizational principles of the brain and spinal cord. This essay embarks on a comprehensive evaluation of the claims articulated within that seminal work. A dedicated exploration of each section will unveil its central arguments, scrutinize the presented evidence, and assess the enduring impact of Cajal's insights on our understanding of neuroanatomy and function. Section 1: General Principles of Histological Technique and Neurohistology. This initial portion of Cajal's work elucidates essential methodologies employed for preparing and visualizing nerve tissue. He expounds upon the virtues of metallic impregnation techniques, particularly the silver staining method developed by Camillo Golgi. Cajal claims that Golgi's method, when judiciously applied, allows for the complete and selective staining of individual neurons, rendering their intricate morphology visible under the microscope. This claim is substantiated through detailed descriptions of the staining procedure, highlighting the crucial role of fixation, impregnation, and subsequent processing of tissue samples. He emphasizes the importance of careful observation and meticulous documentation, stressing how artifacts can arise from improper technique, potentially misleading interpretations. Cajal acknowledges preceding methods, such as those employing Kalmine or Weiget's myelin stain, but asserts they provide limited resolution of individual neural structures, obscuring the full complexity of neural circuits. The assertion rests on the demonstrable ability of the Golgi stain to reveal hitherto unseen details of neuronal operization and connectivity, revolutionizing the study of the nervous system's cellular components. Section 2: Structure of nerve cells and their processes. Cajal dedicates considerable attention to describing the detailed morphology of individual neurons, focusing on their characteristic features: the cell body or soma, the dendrites, and their axon. His primary claim is that these structures represent distinct functional compartments, specialized for receiving, integrating, and transmitting neural signals. He posits dendrites as receptive surfaces, collecting inputs from other neurons. The soma, he argues, acts as an integrative center where incoming signals are processed. He describes the axon as the outboard pathway, conducting electrical impulses away from the cell body towards other neurons or target tissues. Cajal presents drawings and photomicrographs, meticulously detailing variations in neural morphology across different brain regions and species. He observes that neurons are not uniform entities but exhibit remarkable diversity in size, shape, and dendritic branching patterns. This observed diversity, he proposes, reflects the functional specialization of neurons within specific neural circuits. He makes the claim that these differences in structure, combined with specific connection patterns, ultimately determine the unique contributions of different neurons to behavior. Section 3: The law of dynamic polarization. A foundational claim within Cajal's monograph is the law of dynamic polarization, asserting that nerve impulses travel in a single direction, from dendrites and soma to the axon. This directionality, he argues, is fundamental to the ordered flow of information through neural circuits. Cajal supports this assertion through detailed observations of synaptic structures, the specialized junctions where neurons communicate with each other. He describes synapses as points of closer position between the axon terminal of one neuron and the dendrite or soma of another. He notes a consistent directionality in synapse formation, with the axon terminal always positioned presynaptically and the dendrite or soma postsynaptically. This structural asymmetry, he argues, provides an anatomical basis for unidirectional transmission of electrical or chemical signals. Cajal does acknowledge that some neurons may exhibit unusual branching patterns or synaptic arrangements, but asserts that the principle of unidirectional flow generally holds true throughout the nervous system. The validity of this claim rests on the overwhelming evidence of polarized synapse structure and the functional consequences of such polarization for neural circuit function. Section 4 Structure of the spinal cord. In his exploration of the spinal cord, Cajal details the arrangement of neurons and their processes within the grey and white matter regions. He claims the grey matter, organized into distinct laminae, is the site of synaptic integration, where sensory information is processed and motor commands are generated. The white matter, composed primarily of myelinated axons, serves as the communication highway conducting signals between the brain and the periphery. He identifies several key cell types within the spinal cord, including motor neurons, sensory neurons, and interneurons. Each cell type, he proposes, contributes to the specific functions of spinal reflexes and motor control. Cajal utilizes Golgi-stained sections to delineate the intricate branching patterns of dendrites and axons within the grey matter, illustrating how these connections form complex neural circuits. He speculates that the precise arrangement of these circuits determines the specific patterns of motor output generated by the spinal cord. Cajal's conclusions are supported by meticulous anatomical observations and comparative analyses across different vertebrate species. The relative invariance in spinal cord architecture lends credence to the claim of conserved functional organization. Section 5 The retina neural organization and photoreceptor connections. Cajal presents a detailed account of the cellular composition and organization of the retina, the light-sensitive tissue lining the back of the eye. He identifies and characterizes several distinct layers and cell types, including photoreceptors rods and cones, bipolar cells, ganglion cells, horizontal cells, and amacrine cells. His primary claim is that these cells are interconnected in a specific and precise manner, forming a neural circuit that transforms light energy into electrical signals and transmits visual information to the brain. Cajal meticulously describes the synaptic connections between different cell types, emphasizing the role of bipolar cells in relaying signals from photoreceptors to ganglion cells. Horizontal and amacrine cells, he proposes, modulate the flow of information through this pathway, contributing to contrast enhancement and lateral inhibition. He observes, at light striking the photoreceptors triggers a cascade of biochemical events that ultimately lead to changes in membrane potential. These electrical signals are then transmitted to bipolar cells and subsequently to ganglion cells, which send action potentials along their axons, forming the optic nerve. Cajal's descriptions of retinal circuitry provide the anatomical basis for understanding how the eye transduces light into neuronal signals. Section 6 The cerebellum granule cells, Purkinje cells and mossy fibre pa. Cajal dives into the intricate structure of the cerebellum, proposing a model of cerebellar circuitry centered on the granule cells, Purkinje cells, and mossy fibre pathways. He asserts that the cerebellum functions as a sophisticated processor for coordinating movement and maintaining balance. The mossy fibres, he argues, carry sensory information from the spinal cord and brainstem to the granule cells. Granule cells, the most numerous neurons in the cerebellum, send their axons, called parallel fibres, through the molecular layer, where they synapse onto the dendritic trees of Purkinje cells. Purkinje cells, the principal output neurons of the cerebellar cortex, send their axons to the deep cerebellar nuclei, which in turn project to other brain regions involved in motor control. Cajal suggests that the unique arrangement of these cells and fibres allows the cerebellum to perform complex computations, comparing sensory information with motor command to fine-tune movements. He theorizes that the parallel fibres, by synapsing onto multiple Purkinje cells, create a distributed representation of sensory information. The Purkinje cells, in turn, integrate these inputs and generate precisely timed outputs that influence motor behavior. Cajal's model of cerebellar circuitry provides a framework for understanding how this brain region contributes to motor coordination and learning. Section 7 Cerebral cortex layered organization and pyramidal neurons. Cajal turns his attention to the cerebral cortex, the outermost layer of the brain responsible for higher cognitive functions. He describes the cortex as being organized into distinct layers, each characterized by a unique cellular composition and connectivity pattern. He claims that pyramidal neurons, the most abundant cell type in the cortex, are the primary computational units of this brain region. Pyramidal neurons, named for their characteristic pyramidal-shaped cell bodies, have a prominent apical dendrite that extends towards the cortical surface and several basal dendrites that project laterally. Cajal observes that pyramidal neurons receive inputs from other cortical neurons as well as from subcortical structures, such as the thalamus. He hypothesizes that the layered organization of the cortex and the specific connectivity patterns of pyramidal neurons allow for complex information processing. Cajal suggests that the different cortical layers perform different computational operations, integrating sensory information, generating motor commands, and supporting higher cognitive functions such as language and decision-making. His descriptions of cortical architecture provide the foundation for understanding how the brain performs its most complex tasks. Section 8 Neuroglia glial cells. Cajal dedicates a section to neuroglia, or glial cells, often viewed as the glue of the nervous system, though he clarifies that their role transcends mere structural support. He makes the assertion these cells, including astrocytes, oligodendrocytes, and microglia, play crucial roles in neuronal function, providing metabolic support, maintaining the ionic environment, and modulating synaptic transmission. He describes astrocytes as star-shaped cells that surround blood vessels and neurons, regulating the flow of nutrients and removing waste products. Oligodendrocytes, he notes, are responsible for forming myelin, the insulating sheath that surrounds axons and speeds up the conduction of nerve impulses. Microglia, he identifies as the resident immune cells of the brain, scavenging debris and protecting the nervous system from infection and injury. Cajal posits that glial cells are not merely passive bystanders, but actively participate in neuronal communication. He observes that astrocytes can release neurotransmitters and modulate synaptic activity. Oligodendrocytes are myelinating axons, dramatically increase the speed and efficiency of neural signaling. Microglia, by removing damaged cells and debris, help to maintain the health and integrity of the nervous system. Cajal's recognition of the importance of glial cells paved the way for future research into their diverse functions and their roles in both health and Section 9 Development of the nervous system. Cajal dedicates considerable focus to the formation and maturation of the nervous system. He proposes that the development of the nervous system is a highly ordered and precisely orchestrated process, driven by a combination of genetic factors and environmental influences. He describes how neurons are generated from neural precursor cells, migrate to their final destinations, and extend their axons and dendrites to form synaptic connections. Cajal observed that the growth cones, specialized structures at the tips of growing axons, guide axons to their targets by sensing chemical cues in the environment. He hypothesizes that these cues act as attractants or repellents, guiding axons along specific pathways and ensuring that they make appropriate connections. He further asserts that synaptic connections are not fixed but are constantly being refined and modified throughout life. This process of synaptic plasticity, he argues, is essential for learning and memory. Cajal's insights into the development of the nervous system laid the groundwork for the field of developmental neuroscience, providing a framework for understanding how the brain is assembled and how it adapts to experience. Section 10 Pathology of the nervous system. In his examination of nervous system disorders, Cajal outlines the cellular and structural changes associated with various pathological conditions. He claims that these changes, including neuronal degeneration, inflammation, and tumor formation, can disrupt the delicate balance of neural circuitry and lead to a variety of neurological symptoms. He observes that neuronal degeneration, characterized by the loss of neurons and their processes, is a common feature of many neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease. Information, triggered by infection or injury, can damage neurons and disrupt synaptic transmission. Tumor formation, the uncontrolled growth of cells in the nervous system, can compress and destroy surrounding tissue. Cajal suggests that understanding the pathological processes that affect the nervous system is essential for developing effective treatments for neurological disorders. He emphasizes the importance of studying the cellular and molecular mechanisms underlying these processes with the goal of identifying targets for therapeutic intervention. Cajal's work on the pathology of the nervous system laid the foundation for the field of neuropathology, providing a framework for understanding the structural basis of neurological disease. He asserts that future progress in treating these disorders will require a deeper understanding of the complex interplay between genetic, environmental, and cellular. Section 11 Conclusion and Future Directions. Cajal concludes his comprehensive monograph with a summary of his key findings and a discussion of future directions for research in neuroanatomy and neurophysiology. He reaffirms his conviction that the neuron doctrine, the concept that the nervous system is composed of discrete cellular units, is the cornerstone of modern neuroscience. He acknowledges that the neuron doctrine was initially met with skepticism, but asserts that it has been increasingly supported by experimental evidence. He emphasizes the importance of using the Golgi method and other staining techniques to visualize the intricate morphology of neurons and their connections. Cajal calls for further research into the mechanisms of synaptic transmission, the development of the nervous system, and the pathological changes associated with neurological disorders. He suggests that future progress in understanding the brain will require a multidisciplinary approach, integrating anatomical, physiological, and biochemical techniques. Cajal expresses optimism about the future of neuroscience, predicting that continued research will lead to new insights into the workings of the brain and new treatments for neurological diseases. His words, imbued with a fervour of scientific exploration, serve as an enduring inspiration for generations of neuroscientists. The rigorous documentation, the meticulous analysis, and the innovative vision embedded within texture of the nervous system of man and vertebrates secures its place as a monumental achievement in the history of science, a testament to the transformative power of careful observation, insightful interpretation, and unwavering dedication to the pursuit of knowledge. The detailed, section-by-section analysis underscores the enduring value of Cajal's meticulous observations and insightful interpretations contained within the monograph. His meticulous analysis provided the foundations upon which modern neuroscience was constructed, a testament to the enduring power of careful observation, insightful interpretation, and unwavering commitment to scientific inquiry.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.